Hello my dear little champions, how are you all? Let's see if the stream is working or not. We'll just try to load in a bit. Ma'am, can you check once whether it's happening in your mobile phone? Okay, children, just give me a second. I'm not able to load the info up here. I'm not able to see anything. Okay, I think it's live, but still a second. It's working? Children, just give me a second if at all you're watching. Okay, that would be really helpful if you do so, because I am not able to see anything, and that is very much important. How will I see your answers? Yes, just give me. It's happening. Ma'am, can you do me one help? Yes. Can you pass me my laptop? You can remove the charger. I hope it stays here, right? It should stay here. Anyways, the mobile gave up, but the laptop should not give up. If it gives up, that will be a big problem. Okay. Okay, so today there is a big picture. I would be able to see myself in a big screen today. So yes, hi Prajwal, hi Srinivas, hi Shagufta, hi Jyotika, Lakshmi, hi dear. When is 5-marker question discussing? We would be discussing 5-marker question on Monday for biology, okay? Now Girish, hi dear. Mamta, hi. Supreme, hello, hi. Avi Reddy, I hope, yeah? Okay, Ravi Reddy, I'm sorry. Everyone, hi. So let's begin now, since we have already become late to start the question. So what is the rule? We say the answer by writing like this: 1 A, B, C, D. So earlier to the commercial production of human insulin, insulin was obtained from. So now it is being commercially produced by using our genes and everything. But initially, okay, before so many years, how was it produced? From the camel or the pig, and sheep and the cattle, pig and the cattle, dog and the pig. What is the answer for this particular question right over here? Shagufta says 1C. Hi Pranavi, that's someone new over there. Hi Makbul. So 1C. Srinivas also says 1C. Girish, Mamta, all of you are saying C. So let's log C, because your answer is actually correct, and that's so kind of you, Aishwarya ma'am. Thank you for putting my laptop on charger. So yes, early to the commercial production of human insulin, insulin was obtained from pig and the cattle. Okay, now it is not being used. Why? Because it can cause allergy in our body. So yes. Going ahead, second question. Over 95% of all existing transgenic animals are mice, sheep, pig or rabbit. Transgenic animals as in we are putting our genes into them and making them produce a lot of things, or probably putting our genes and testing what are the actual scenarios, what does it code for, and things like that. So these are transgenic animals, my dear children. Lakshmi says 1C, Prajwal says C's. Okay. Prajwal, hello. Shagufta says 2A, Jyotika 2A, Lakshmi says 2A, Srinivas 2A. Oh my god, lot of people A. Mamta, Pranavi, Prajwal, Gayatri, hi. So all of you are saying A, which is why I would lock A. Yes, 95% of all existing transgenic animals are, my dear children, mice. You know, mice and us have a lot of things in common. Okay, almost. Okay, a lot of our DNA is kind of same. We are kind of little bit different, and that's how I became a human, or you became a human, and that became a mice. So you see that nature. So next thing, humulin. Question number 3. A genetically engineered insulin was produced for the first time by Biotechnology Limited, Glaxo, Eli Lilly and Company, or Cipla. So Srinivas is asking, ma'am, these MCQs are enough? These MCQs would definitely solving a lot of your problem, and probably if you're lucky enough, these questions would be directly appearing on the day of exam as well. So, but yes, MCQs, no one can take the guarantee of it, but with MCQs, you cover a lot of concepts, and from those concepts, you get more MCQs. So that is why it goes on. Yes, so a lot of you are saying answer. Shagufta again, hi 3C, Gayatri, a very good evening dear. Srinivas 3C, Pranavi 3C, Gayatri 3C, Mamta 3C, Netra, oh my god, answer is C, ma'am. Mangalappa is also here, hi dear. So all of you are saying, which is why I will be locking Eli Lilly and Company. This is a very old company, it's still there in India, and yes, it is the first one that used genetically engineered, that helped in the production of genetically engineered insulin with the help of which particular microorganism, my dear children? Do you know which particular microorganism was the one that helped in the production of the insulin? That can also be a question. So Srinivas said, is this MCQ enough? So this is how MCQs are being formed. So there was an organism, microorganism, with the help of which we produce the genetically engineered insulin, that is humulin. I want to know the name of that. So Prajwal, you're waiting. Hi, so I'm so sorry. So you're waiting for the video. Yes, which video? Can you just tell me that? So there has been a 1 marker question that is going there. So you were waiting for my video. Oh, that's so sweet of you, Prajwal. Okay. So this is the answer, but can you tell Shagufta, Pranavi, whoever is there, okay, Mamta, can you tell the name of the microorganisms which was used? Come on children, this is very much important. If you are not telling the answer, probably I would only have to tell the answer. Yes, it is E. coli. So E. coli was used where our gene was taken and put inside the E. coli, and this E. coli later produced the protein that we were looking forward for. Okay. Now going ahead with the 4th question, conversion of Bt inactive toxin into active form requires. So what does this question mean? Conversion, yes. So Shagufta, Pranavi, Mamta, Prajwal, Girish, Mangalappa, Aman, hi Aman. Everyone is correct. Okay, children, can you tell me the answer for this? So this question says the Bt toxin that is produced by Bacillus thuringiensis, no? It is usually inactive toxin, but it becomes active. And how does it become active? Does it need alkaline pH or acidic pH, neutral pH or high temperature? What is the answer for this? So a lot of you are saying, okay, A. Let's see. Alkaline pH, that's what you are saying. Gayatri, Shagufta, Srinivas, Pranavi, Mamta, Mangalappa, Girish and Pradeep. All of you, hi Pradeep, all of you are saying A. I saw just one answer with C. Baba, C is not the correct answer. A is the correct answer. It needs alkaline pH. And how does it get it? Basically, suppose I am a bacteria, I am producing this toxin, but in my body it is inactive. Now, one particular insect came and ate me. Now, when I get into the insect's body, my dear children, in their gut there is alkaline pH, and now the toxin will become active toxin, and it is going to affect certain cells of the insect and going to kill it. The certain cells which are there, it will be coming right. That is the next question probably, so I'm not going to tell you right now, but it is alkaline pH. So whoever got alkaline pH, you all are correct. Okay, Prajwal, I hope I was able to explain you the answer. Okay. Chalo. Next, yes, the activated Bt toxin binds to the surface of the cells of the midgut of insect. Which cells of the, I told you, it goes up into the gut of the insect and gets activated there and is going to damage certain cells. So do you know the name of the cells? Okay, I captain, that's great, so you got it. So epithelial cells, Campbell cells, nerve cells, blood cells. Ma'am, will this MCQ help in CET? Yes, definitely. MCQs, whatever MCQs you're going to solve from any chapter, my dear children, it is going to help in your KCET exam. Okay? So Shagufta says 5A, Gayatri says, Pradeep said 5A, Gayatri, Girish, Pranavi, Mangalappa, Mamta, M. Girish, oh my god, a lot of you are saying, okay, epithelial cells of midgut, not midgut of epithelial cells, okay. My dear children, epithelial cells of the midgut. Midgut is a region, right? Now in this region you have epithelial cells. So those cells are attacked with the help of this toxin. So your answer is A. Epithelial cells of the midgut, okay? Do not get it wrong. Hi Revana. Chalo, going ahead. Question number C6. C-peptide is what? C-peptide is a part of mature insulin, not a part of mature insulin needed for activity of insulin, reduces the blood sugar level. 17 people are watching this video, then why I am saving only 7 people up over here? Where are all the 17 people? My dear children, please put down your answers. I would be just happy to see your answer, whether it be wrong or right. So just tell me the answer. We are here to be friends and to study a lot of important things. Right, so do not feel the fear. Jasmine Ma'am is very papa person. I'm not going to do anything. So, yes. So, Mangalappa 6B, okay. Revana 6B, Gayatri, Lakshmi, hi Akash. Good going. Lakshmi, Mamta, Girish, Gayatri. If at all I'm missing some names, please forgive me, okay. Pradeep and Shagufta already said B. So, yes, C-peptide is not a part of mature insulin, exactly. So, you know, insulin is produced as a pro-hormone. That means it is not a proper hormone as of now. So, it has A peptide, B peptide, A and B peptide, and to link this, they also have C-peptide. So, now this is known as pro-insulin. It is a pro-hormone. It is not very much active. Now, what happens? In the eukaryotes, this particular C-peptide is being cut, is being removed, my dear children, and then later the A and B which is there, it is being joined, and that's when you get the actual version of the mature insulin. So this becomes your answer for the question. I hope you got that. Okay, Prajwal, Reddy, every one of you are correct. Kudos to all my children. Okay, going ahead with the next question. ADA is required for A. Proper functioning peptide. Ma'am, what is C-peptide? Listen, peptide are nothing but group of amino acids which come together. So, you know, insulin and everything over here, my dear children, they are made of small protein units. So, over here, this is A peptide, as in this would be made of some amount of amino acid. B peptide, again made of amino acid. C, some other amino acids. So, you need to remove the C peptide and join A and B with the help of disulfide bonds. And that's how you make the active form of insulin. Okay, now yes, where was I? Yes, proper functioning of immune system, maintaining optimum oxygen content in the blood, maintaining the blood sugar level, synthesis of clotting factor. What is the use of ADA? What is ADA? Anyone with the full form of ADA? My champions, anyone up over here with the full form of ADA? Hi Gayatri. So I see new people up over here. Shagufta, Pradeep, ma'am, video was not clear. Is it clear now? Revana, Mangalappa, Pradeep, Pranavi. Hi Pranavi. So every one of you is saying 7A and it is the actual answer. ADA actually stands for adenosine deaminase, which is an enzyme. And obviously this particular enzyme is very much important for the proper functioning of the immune system, my dear children. Yes, deaminase. Good teacher and good teaching, ma'am. Thanks to you, Prajwal and Pradeep. Great. Gayatri, great. Pranavi, great. All of you are correct. Just go with this, check the spelling once, okay? Shagufta. Okay. Going ahead, this is the answer. Akash, can I just use the screen? Yes, just a second, okay? Okay, next question. Nematode resistant genes are transferred to plants with the help of, as in a plant was getting affected because of a worm that is a nematode. Okay, nematode is basically, we call it a category of worm as helminthes. Okay, now you want to make the plant resistant of this particular nematode. So you insert certain genes. So how do you insert this particular genes into the plants? So you should be knowing the plant vector. Then only you would be able to answer it. So 8A, Pradeep says 8A, Prajwal, Gayatri, Bhumi, hi Bhumi, Lakshmi, Mamta, Srinivas, Mangalappa, Pranay, all of you are correct. Just that person who gave C answer, can you just recheck it once? You are going really, really great. Just check it once. It will be all right. Okay. So, nematode resistant genes are transferred to the plants with the help of Agrobacterium, my dear children. Now, this is a bacteria. Its whole name is Agrobacterium tumefaciens, and it is the usual vector for transferring genes into the plant. So, what happens is that this Agrobacterium tumefaciens is a villain actually. So, it has a plasmid which is known as Ti plasmid, and the Ti plasmid has a region that is known as tDNA. Now, when the bacteria affects the plant, it passes on this tDNA into the plant, and this tDNA causes tumor. Now, we are going to use this machinery for our own use. How? We are going to put the gene of interest over here and remove this tDNA. So, there is no tDNA, but just our gene of interest. Still, it will be transferring. It does not know that we have cheated it. Okay, it still will be transferring the thing. So, that's how the plants become resistant. Okay, yes, it is the cause of tumor in dicot plants. So, your answer should be A. Okay, I hope you understood it, children. I am assuming you understood it. So, if you have understood, can you just put heart or some, like or whatever you want over here, okay? Or you can just say it's clear, either way. Yes, ma'am, got it. Chalo, great. That's the best way to answer. So, Nitya. So, ADA is caused due to, why do you get ADA? Hi, thank you, Pradeep. Why do you get ADA? Deletion of a gene coding for adenosine deaminase, addition of a base in the gene of adenosine deaminase, removal of base, okay, or shifting of one fragment. All of you have said heart. Thank you so much, thank you so much, thank you so much for all this and this. Thank you for acknowledging. And can you please tell me the answer for the 9th question? So, any one of you, if you feel that you are not able to answer the question, it's totally fine. See your friends who are answering the question, okay? Understand what I'm trying to tell, rectify the mistake, and that's how we learn. This is how we learn certain new things. It's totally fine to get it wrong. So, ADA is caused due to so, a lot of you have answered starting with Mr. Artist, Pradeep, Gayatri, Srinivas, Shagufta, Prajwal, Mr. Artist, Mamta, Lakshmi, Akash, Bhumi, Gaya. This time Bhumi got it correct. Gayatri, Nitya, Pranavi, every one of you are saying A. Answer is A, and this is a direct question from your NCRT. Adenosine deaminase is caused by deletion of a gene that codes for this particular enzyme. This enzyme is a protein, and genes codes for protein. So, if you delete that gene, this particular protein would not be coded. So, your answer is A. Cool. Next question. In the gene therapy for ADA deficiency, functional cDNA is introduced into. What do you mean by functional cDNA? cDNA as in complementary DNA. Where do you get the complementary DNA from? So, you know, you take this DNA which do not have any introns, okay, they do not have any non-coding regions. This will be just coding for the thing that we are looking for, that is ADA. Okay, this is the complementary DNA. So, what are we doing in gene therapy? In people who do not have ADA, we are going to insert this particular DNA. But where are we going to insert into? So, Pradeep says A, Shagufta says A, Pradeep, Mr. Artist says A, Gayatri A, Srinivas, Nitya, Prajwal, Gayatri, okay, Mamta, Bhumi, Akash, everyone is saying A, Lakshmi also. Anyone to challenge them? All of them? Kudos, my dear children, all of you. I am so happy to see you answer, and you all are correct. Yes, it is lymphocytes. So, basically what is done is that, yes. So, basically what is done is that in my body, suppose I am a person who does not have ADA. So, from my body, the lymphocytes are being taken out. Okay, and in this lymphocytes, this particular cDNA has been inserted. Then what do? You take this lymphocyte and put it back into the body. You put it back into the body. So, there will be certain lymphocytes with the ADA, and the functioning will be all fine for some time. But obviously, you have to do this again and again. Okay, you have to do this again and again. So, your answer is lymphocytes, not any other thing which is up over here. Yes, enzyme replacement therapy. Okay, going ahead. GM, golden gene. Genetically modified golden gene is enriched with vitamin. Now, this question you would be able to answer only if you have read your NCRT properly. Okay, golden rice, if you know. Okay, there are so many people who read the NCRT properly or what? Oh my God. Ranavi says A, Pradeep says C. Okay. Mr. Artist says C, Shagufta says A, Gayatri says A. Okay, come on children, tell me all the answers. A, B, C, D, whatever you want to say. Srinivas says D. There are only A, B, C, D option, okay? Don't give me E option over there. Hi Sanjay. So someone says K also. Okay, it's totally fine. Vitamin C is not there. So the answer for this question is A. I'll tell you one thing. So people, okay, remember, okay, C option is the correct answer. Why? I'll tell you. So people generally in Africa and all, they had didn't have very good diet and all. So they had very poor eyesight. And you know, for improving eyesight, what do you have? Vitamin A is playing a major role in eyesight. So for at least having good amount of vitamin A, this particular rice was introduced to them also, so that they can have this rice and improve their vision. You get it? Now you would not forget it. So A is for eyesight and golden rice, and rice was eaten by a lot of people, right? So obviously thought of making golden rice, which was rich in vitamin A, my dear children. So will you forget this? The answer is C. Okay, please do not forget this. I hope it is clear. If it is clear for all of you, give me a thumbs up and then we would be going ahead. Why did you guys got stuck, or did I get stuck? Okay, Pradeep gives me a thumbs up. Okay, I assume that, okay, this is a slow process that happens, which is why sometimes I do not get a track whether you are able to understand or not. So Shagufta also gave. So thank you for acknowledging what I just said. Now we'll move on to the next question. The answer for this was that, okay. Teju, hi dear. Prajwal, thank you. Srinivas, thank you. So which nematode infects the roots of tobacco plant? So I told you nematode is nothing but a worm, Ascalmentis. Okay. Thank you Sanjay, Rayan, everyone. Gayatri is so sweet of all of you. Now which nematode infects the roots of tobacco plant, my dear children? What is the name of that? You have Meloidiga incognitia, Ascaris lumbricoides, Entrobius vermicularis, Becqueria bancrofti. So human health and diseases might be coming up over here. Please don't get confused. So everyone okay, ma'am, India needs 230 runs to win. Either you win over here right now. We will think about that. See what is happening in this country. So 12A, Shagufta, Mr. Artist, Shagufta, Gayatri, Srinivas, Rayan Khan, hi, Pranavi, Akash. Every one of you correct. Okay. Prajwal, Jyotika, Gayatri, Rayan, Mamta. Okay, so all of you are saying A. Yes, the answer is A, that is Meloidiga incognitia. This is going to affect the roots of the tobacco plant, and against this a new technique was seen. Not a new technique, basically something was sorted. I will be talking about that in the next question. Yes. Which one is the method of cellular defense in animals and plants? Recombinant DNA technology, immune response, RNA interference, demutation. Which one of them is a cellular defense? Okay, the cell only tries to defend itself in the case of animal and plants by this particular method. What is that particular method? So, the Meloidigan infection that was caused to tobacco plant, that was also made fine with the help of this only, this method. So, if you know, you know. Okay, so children, please answer like whatever question you're saying. So 13th is C is what you're saying. Okay, 13th is C. Shagufta says B, and Pradeep, RNA interference is what you're saying. Rayan says C. Okay, Prajwal B. 13C, B, RNA interference. Children, okay, I'm so sorry for that. So this is B up over here and C up over here. That is why you got confused. Okay. So take RNA interference as B option only. Okay, as B option only. So your answer is correct, my dear children. This is a method of defense in the case of animals and plants, and I'll tell you how. So basically, if any organism try to infect us, mostly if it is a, anything, virus, bacteria, whatever is there. So it basically has its RNA. This mRNA, my dear children, has all the information to make proteins. And now this protein can be some toxin or whatever is there. Now we do not want this particular organism to make the protein in our body. What do? We do a technique that is known as RNA interference, where what is happening is that a double-stranded RNA is introduced. A double-stranded RNA is introduced, From here, one particular strand will make a complementary strand with the mRNA. And if there is a complementary strand with the mRNA, my dear children, it would not be undergoing the process that is known as translation. And hence, it won't be able to make the protein. And tada, we are safe. So, the process of silencing of specific RNA due to complementary double-stranded molecule binds and silence the pest RNA. Exactly. So, your option is B, RNA interference. Okay. So, this has been, that's my fault. Okay. So, I just said, now it's fine. Now it's fine. So, B, everyone who went for RNA interference, okay, you all are correct, whether it's B or C, whatever you said. This is the correct answer. I hope you got it. So, it's for animals and plants that they do it. Cool. Now next, 14th question. Sense and antisense RNA are to each other what? Similar, same, complementary, different. Hi Nitya. Hi Teju. Hi everyone. Okay. Can you tell me sense and antisense RNA are to each other what? Let me see ABCD correct or not. Yes, it is correct. C. Pradeep, Mr. Artist, Srinivas, Shagufta, Gayatri, Girish, Pradeep, Bhumi, Anand, Akash, Ryan, Pranavi, Mamta and Sanjay says complementary. Yes, exactly. So, sense and antisense RNA are basically complementary to each other. So, that is how the RNA interference method works. It will be binding with the sense RNA. Okay. Now, low concentration of any pathogen can be detected by dash of their nucleic acid. By dash of their nucleic acid, or basically you can rather, I will be reframing the question, okay? By detected by which method? By which method? Low concentration of any pathogen can be detected by which method? So, if there is pathogen in my body, but the amount of this pathogen is very less, how can I detect it? With the help of what? Shagufta says 15B, Mr. Artist says 15B, Girish say direct answer, PCR, okay. Mamta 15B, Srinivas 15B, what about others? Sanjay denaturation. Okay, it's okay. Keep on guessing. Gayatri says B, Bhumi 15 PCR, Girish PCR, okay. I see a D option also. Bacha log, it's not denaturation. What do you mean by denaturation? Basically, you are separating the DNA strands. You do not need that. So, over here, low concentration of any pathogen can be detected by a method that is known as PCR. Why? Because in PCR, what do? You have the DNA. You basically create copies of this DNA. So, if I am having just one strand of DNA, that would not be enough for me to identify whether the pathogen is there. But if I am able to amplify it, I am able to increase its copy, then it's easy for me to check, that whether this particular organism is infected by some amount of pathogen or not. So, PCR is the answer, okay, and it is by amplification method that this is being done. Didn't understand? I will explain again. Listen. There is very low concentration of some pathogen in my body. Now, there is a method by which I can still find it. What can I do? Suppose I take my blood and give it for testing. Now, the blood which is there, inside that basically with the help of PCR, the DNA and the genetic material which is there, it is amplified. And when you amplified it, even the small traces of things get amplified, their number increases, and I will be able to spot that, hey, I find a DNA that is not of human being. This is some pathogen which is in there. So, it is with the help of PCR machine, which is why I can help find it. Okay, now I hope it is clear. If it is clear, just give me a thumbs up. Hi, Vikas. I hope you got the answer. This is the answer. Let's go ahead. 16th question, my dear children. 16th question says, ELISA is based on the principle of reaction of what? ELISA, is a test for that we used for HIV and all. Okay, apart from that also, ELISA is being used for other things. But yes, Prajwal, aye, Captain. Okay, clear. So, it is on the principle of reaction of what? What is reacting in the case of ELISA? Enzyme-linked immunosorbent assay. What plays a major role over there? Pranavi says B, Pradeep says B, Vikas says C. Okay. Girish, okay, thank you. Amit, hi. Pranavi says Gayatri. Okay. I see a mixed answer over there. Akash, Vikas. Okay. Children, Your answer is antigen-antibody binding. Okay, this is the thing. So basically what is being done is that, suppose there are different ways of ELISA, okay, there are different forms of ELISA. In one form, what is done, the antibody is being already put over here. We know what is this antibody for. We know that this is being released only if some person is affected with this particular disease, some disease, for example, HIV only. Okay, now this antibodies are specific for HIV. Now I want to test whether one person has HIV or not. So what do? I take their blood, okay, and then I would be having antigens. Now I would be putting these antigens up over here. Now if these antigens are going to bind with these antibodies, my dear children, that means what? That person has HIV, which is why basically they are able to bind with the antibodies which are specifically produced against HIV. But still I cannot see it. How do I know it? So what do? You put next set of antibodies, and these antibodies again are the ones that are produced in respect to HIV only, and you are going to tag it. You tag it with some substance, from chromogenic substance, some coloring substance, okay. Now these antibodies will also be binding to these antigens. These antibodies will be coming and binding to these antigens, and they also have coloring substances. Now when I add the substrate, my dear children, the color changes. The color over here changes, showing that yes, this whole reaction has taken place. The antigen, antibody, everyone has bound to each other. That means this person will be having what? This person will be having HIV/AIDS. Okay? I hope now you got it. Guys, please concentrate here. Okay, I hope you got it. This is ELISA, which is based on the principle of antigen-antibody reaction. Your answer will be C. Cool? Well, this is a kind of confusing thing. So if you have understood, do let me know. Okay, shall I go ahead? If I can go ahead, put a mark like this, then I would be going ahead. Children. Yes, Sanjay. Okay. Let's go ahead then. 17th question, a very direct question from your NCRT. That is, India alone has dash varieties of rice. Thank you, Vikas. Thank you, Sanjay. Thank you, Pradeep. Everyone. And I hope all the other children got it cleared. Thank you, Gayatri. Thank you, Amit. So what is the answer for this? Thank you, Shagufta. 17th question, what is the answer for this? India alone has dash varieties of rice. 1 lakh, 2 lakh, 20,000, 27. What is the answer for this? Shagufta 17A, Pranavi B, okay. Gayatri C. Guys, don't give me an option, no. Nirmit 27, okay, Okay, so I got all the options, so it's an MCQ question for me, right? So my dear children, again it's a straight question from your NCRT. India has up to 2 lakh varieties of rice, my dear children. So your answer is B. Your answer is B. Okay, this is given where, no? Towards the end where there is patent and biopiracy and stuff like that, no? You will be finding this question. Okay? So your answer is B, 2000. So 2 lakh. So there are estimated 2 lakh varieties of rice in India alone. This is given on page number 185 of your NCRT textbook. Going ahead with the next question. So I hope all of you got it correct. This is the answer. Now, again a very direct question. You might be missing this, but it's okay if you miss it. Okay, but you still have to keep guessing. Now, the Indian Parliament has recently cleared the dash amendment of the Indian patent bills. 8th amendment, 1st, 2nd, 3rd. What is the answer for this? Thank you, Pradeep. Thank you so much. Means a lot. Children, come on, buckle up. You can do it. You can open your NCRT and try to find the answer for this. I do not mind. Open your NCRT and try to find the answer for this question. The Indian Parliament has recently cleared the dash amendment of the Indian Patents Bill. In that way also you remember. Mr. Artist says 18D, Shagufta says C, okay, A, D. Okay. Pranavi says D. Mishtartis says D. Bhumi says C. Grish says D. Srinivas says D. Grish finally said it's second. Okay. Children, the answer is second. The answer is second. So it's given again in your NCERT. Do you want to know where in your NCERT? I'll be telling you on 185 page number, the, what do you call it? The fourth line. Okay, the fourth line towards the end, last para, the first line, sorry, the first line which is there, my dear children, in the last para, there you get the answer. It is second. So you should be going to details of NCERT. You see, this is how the question is being asked. Now, going ahead, this is not to scare you, but to make you aware of certain things, that's all. Now, today transgenic model exists for diseases like, 19th question, today transgenic model exists for diseases like, so we have models for diseases such that basically we can try drugs on them and see whether it's affecting or not. Okay? Exists for diseases like A. filariasis, B. influenza, C. cystic fibrosis, D. none of the above. HET Sunalist, please ask me the doubt, Baba. Tell me. So everyone is saying C. Let's see whether it is C or not. Hi Nirmit, I've become so happy when new people try it out. Okay, that does not mean the old people who are there, I don't feel happy for you. I feel equally happy for you as well. So all of you try, that's all. Ayo. So your answer is yes, cystic fibrosis, and as Pradeep was saying, cancer, cystic fibrosis, Alzheimer's, all are certain diseases for which we have transgenic models nowadays. Okay, so that we can try drugs and find a cure for it. Cool? So your answer is C. All of you who got it C, it's correct, and all of you who got a wrong answer, you are now understanding and now you won't be repeating the same mistake. Did I show you the answer for this question? But it's okay. Select the correct match. Gene Cry1AC, that is for cotton bollworm, Cry2AB for corn borers, Cry1AB for cotton bollworm. What is the answer for this? Select the correct match. No. It's just biotechnology and its application. So apparently what happened, I'll tell you. So I was planning to take biotechnology principles and processes, but I gave the questions for typing and that chapter was biotechnology and its applications. So at last minute I had to change it. So I'm so sorry, but I will be covering biotechnology principles and processes as well. So I hope you forgive me for that. Okay? And today was a very hectic day for us with so many things happening. So, hi Sumadwa. So, you're saying C, Gayatri, Mr. Girish, Pradeep, Shagufta, Amit, Akash, Pranav, everyone has a different answer. Most of you are saying A, and some of you are saying C. So, my dear children, you are asked to find the correct match. So, listen to me carefully always. If you get it, if you listen to me now, you might get easily good marks in this particular chapter because this question is repeated in so many formats. So, the genes are there for toxins, right? Now, you have Cry1AB and Cry1AC and Cry2AB for cotton bollworms. How do you remember it? So, the first letter starts with C. So, first letter starts with C. The second letter starts with B. The second letter starts with B. Okay. So, you might be saying corn borers is also like that, no? But you see, Cry-AC, Cry2AB, basically it's a big thing. Similarly, cotton bollworms is also kind of big. So, this one goes for them. Now, the next one is Cry1AB. Cry1AB is for corn borers. Okay, corn borers, this is only one particular thing for corn borers since they are very short. You can remember like that. Okay, works. Chalo. I hope that that has been done. Let's go ahead. Children, you got it? So your answer is only A. Okay, so there is cry 1AC for cotton bollworm, and this is the only correct option, so this is the answer. Okay, ma'am, do you speak Kannada? No, I do not speak Kannada, but I am in a learning phase. So, nanu Kannada kalita idini. Yes. Chalo. Question number 21, my dear children. Which of the following statement is not correct? Over here, I hope children you understood the other one. Okay. Now, which of the following statement is not correct? Most Bt toxins are insect group specific. Okay, that means they are going to affect only certain insects, certain toxin for certain insects. Now, RNA interference takes place in all eukaryotes as well as prokaryotic organism as a method of cellular defense. And then the recombinant therapeutics do not induce unwanted immunological responses in humans. And then adult onset diabetes can be controlled by taking insulin at regular time intervals. Now, you had to tell me what is not correct. Okay, this is the question. So, Girish says B. Okay. Pradeep says it's C. Gayatri says it's C. My dear children, can I just check the question once more? You are asked what is not correct. Okay. What is not correct? You are saying C. So, let me read C. The recombinant therapeutics do not induce unwanted immunological responses in human. Obviously, it should not induce, right? If it induces, can we actually take it safely? No. So, this is correct statement. You are asked about what is not correct. Okay, you are asked about what is not correct. So, your answer should be B, my dear children, because RNA interference takes place just in eukaryotes but not in prokaryotes. This is a wrong statement. We read that in animals and plants this RNA interference and everything takes place. Animals or plants are what? They are eukaryotes. They are not prokaryotes. They are eukaryotes. So, your answer becomes B. This is not correct. Okay? Works? Can we go ahead? Children? Okay, 21, please correct it to B. Cool. Now, which of the following statement is incorrect about gene therapy in adenosine deaminase deficiency? What is again incorrect? So, please spot incorrect word and then term. Now, lymphocytes from patient's blood are taken out and cultured. A functional ADA, complementary DNA, is introduced into these lymphocytes. Lymphocytes are then reintroduced into the body of the patient, as in they are put in back into the body of the patient, and patient does not require periodic infusion of genetically engineered lymphocytes. So, what is the answer for this? Shagartha says D. Amit, what is the next chapter? Baba, next chapter would be biotechnology principles and processes. Okay. How many questions? I think you have just few more. Let me see. I forgot how many. I think 24. Okay, 2 more questions. Okay? Works? So, all of you are saying D. Okay, so is it actually D? Yes. Because patient does not require periodic infusion would be totally wrong. Definitely patient do require periodic infusion because it is not completely cured, right? How can you completely cure this particular disease, that is ADA, by only one method, that is in the embryological stage only. If you are giving the adenosine deaminase, having DNA or cells having this particular gene particularly, that is from the bone marrow, you take these cells and now you insert it into the embryological cells, okay, embryological cells, and then that's how you can treat it completely. But obviously in the embryonic stage, no one can easily find out the disease, no one predicts this, which is why they do not know it. Okay, so now I hope this is clear. Your answer is D, and with this we go ahead. Now, question number 23. Okay, ma'am, next chapter is biotechnology principles and processes, and it will be on next Thursday. Okay, works? Children, are you liking this video? Please, if you're liking, do not forget to like the video as well. Put down the comments in the comment section, and definitely do not forget to like and subscribe to our little channel. So, match the column 1 with by Prajwal with column 2 and select the correct option from the given quotes. Now the second last question. Alpha-1 antitrypsin you are having. Okay, now alpha-1 antitrypsin is being used to treat what? Treat one particular disease, that is emphysema. Emphysema, okay, which is a respiratory disease. Now going ahead, you have transposon. What are transposons? Transposons are the one that can basically move from one area to another. These are genes and they can move from one area to another. So they are mobile genetic elements, my dear children. Now you have ELISA. ELISA, as I said, is a test for checking AIDS, HIV, yeah, jumping genes, exactly. Now retroviral vector. Why do you need it? Retroviral vector, you use it in gene therapy. Why? In gene therapy, you are basically doing in human beings, my dear children. And in human beings, what is the usual vector? It is retroviral vector only. So your answer for this question will be, you just have to check D. Retroviral vector goes with gene therapy. So for D, it's 2. And only one option has D with 2. So let's see. A goes with 3, correct. B goes with 4. C goes with 1. And your answer becomes B. So everyone who said B, my dear children, Pradeep, Girish, Akash, Shagufta, Pranavi, Srinivas, Gayatri, every one of you, great. And Prajwal, all the best to you. Okay, and thank you so much, Savita, means a lot. Chalo, going ahead. The last question right over here, my dear children, that is, read the given paragraph and identify X and Y. X is the right granted by a government to an inventor to prevent others from commercial use of his invention. Okay, it is a right. Now, to prevent others from commercial use of his invention. Now, when X are granted for biological entities and for products derived from them, these are called dash. So, X is a right given to them. What do you call this? What do you call this as? So, someone says A. Girish says A. Are you sure, Girish? Just go through the question really once more, read it peacefully. Okay, Pradeep says C. Shagufta says C. Akash A. Okay, Gayatri C. Pranavi A. Children, can you go through this question? Akshay, hi dear. Children, can you go to this question really once more and see for the option? What is the answer for this? C is the answer for this. Okay, X is patent. Okay, it becomes patent. I only have the right to use it and everything. Now, this whole process of giving this thing is known as biopiracy. It is known as biopiracy. It's not patent. Okay, so your answer is X is patent and Y is biopiracy. With that, we get the answer. Hi, Ashwin. Hi, Radha. So many people up there towards the last thing. So I hope all of you got this particular question, my dear children. Okay, so you know, people were using the rice was produced from our country there. So then we had to tell that, hey, we have the patency for it. You get it? So that was something. And with this, we complete biotechnology and its application. So, my dear children, before going, I want to say thank you to all champions who participated wonderfully in this particular session. I'm really proud of you all. And I have given the model question paper 1 solution. Just go through it. It is very important. All the 3 question papers would be really important. There are 1 marker questions, all 1 marker question solutions which I am giving right now. It's a premiere video. Okay, it's not live, it's premiered. So, we basically see your chat definitely, but we cannot answer it like live. So it is being premiered. So please go and watch it. It's very important for you guys. And Gayatri, thank you so much. Srinivas, thank you. Yashwin, thank you. Shagufta, Pranavi, Amit, every one of you. Bye, my dear champions, and have a wonderful day. Lots of love and take care. Do not be worried. Shagufta, thank you. You guys were also marvelous. So, when is the next chapter? When is the next chapter? Would be on Thursday. And do not forget to like the video and comment and also subscribe to the channel. Bye-bye. Take care. Good night.